let's begin Module 1, Molecular Control of the Cell and the Technologies that Measure the Modulations. So really, this is what you folks are providing. You're providing a tool or a set of tools that deliver on personalized medicine. We've all heard about it. It's everywhere. Personalized medicine. You've got screening through diagnosis, prognosis, and indeed the personalized therapy that, knowing what your molecular profile is of your cancer, will direct the physician to provide the right therapeutic. And you'll hear me talk, and I'm sure you've heard of talking about companion diagnostics. Big Pharma is moving towards companion diagnostics. They have so many of these drugs in the pipeline, yet they can't get them out because if they follow the traditional route, it basically doesn't work. They end up with a lot of failures. So companion diagnostics is a new tool identifying a test that has a molecular profile that is efficacious for the right drug. And then, of course, you can use that for monitoring the disease as you go through with the drug working efficaciously and allowing you then to know if you're going to fall out of remission into relapse. So a good overview of what you folks are providing from a personalized medicine standpoint. And so when you look at the levels of organization of the human body, it's amazing. You start off with the atoms, through the molecules, the organelles, the cells. 50 trillion cells sit in each one of us. That's a lot of cells to begin with. Those connect to organs, to make systems, to make the full human organism there, which is riddled with these letters, which we'll eventually get to, the genetic code. And each one of those cells in itself is like a city or a town or a village within the US, if you like. And it's a powerhouse of everything in there that controls its everyday life, and how each one of these cells connects with its next door neighbor, and how at times things go awry. And when they go awry, that cell then starts to say, I don't like you guys next door to me anymore, I'm going to build my own city. And that's how cancers evolve. And you can see there's a whole load of components in there. We're not going to go through each one of them. We're really going to talk about how the mechanism of a signal from the outside gets to the inside and back out again, and how when that goes awry, that's how cancer evolves. And so the only way in is through this cell membrane. And coming back, if I can come back, this cell membrane here is really the passage in. It's almost like a castle with its walls around it. And it's really made of a phospholipid bilayer. And our next slide describes a little bit more about it. But the way in is usually through, these protein components. They're gates. And this is the one that really focuses around cancer. And it basically means that something comes in, hits the top of it, it has a conformational change, the signal goes inside, and then the message is delivered through, much like a relay race with a baton. And then the other ones that are here, which we won't talk about today, really talk about other ways of getting a signal in. For example, this one here is really about getting ions in and out, calcium, potassium. This one here works in your neurons and is a voltage gated and will explode the components like calcium over to the next cell, and hence I'm able to move quickly. All these explosions are happening really fast. And then you've got one for transporting big proteins, and then the last one is one where the cells talk to each other. They actually do connect, they do talk, and if viruses, bacteria come along, there's all sorts of molecular mechanisms that trigger off certain messages in to start to change the cell, to get proteins out to defend the cell, the organism, and the whole body. So here's the cell membrane. Really nothing to be scared of. It's just a very simple hydrophilic head with a hydrophobic component below it, and simply put, it stops things from coming in and going out, unless of course it's got some passageway, a ticket through to the center and the ticket through back out. It's the phospholipids, as I mentioned. The hydrophilic part attracts the water, and the hydrophobic is naturally not attracted to the water. And as I mentioned, you can't get in or you can't get out unless you have a ticket to ride in or out, and we'll talk a little bit more about that. So here's passing through the membrane. As I mentioned, there is the opportunity for small components to get through under this diffusion. It's a gradient where it's either strong on the outside and weak on the inside and it pulls things through, or it's the other way around, or it's just through passive diffusion. And so, as you can read, hydrophobic particles can pass through, but very large ones only pass through but the specific channels. So small, really tiny molecules can get straight in. Bigger ones need to have some help to get through that phospholipid bilayer. So the overview is very simple. As you can see, you have a molecular mechanism that comes in from the outside the cell. It triggers the receptor, and then through a relay mechanism goes all the way into the cell to the DNA, and then kicks the DNA off to make more protein of a particular type that's required to address this signaling mechanism or molecule that came in. And so for example here then, a hormone could come in and it slides through because it's small enough to do that. And in this example, you have a situation where it's bound to a receptor internally and it's done the same thing that I mentioned before. So you've gone from a molecular mechanism on the outside that needed a protein to be triggered to pull that in or to release a set of mechanisms to something that just slides in and triggers off a receptor on the inside. It goes to the DNA, makes more DNA of a particular type that this is calling for to help you address the mechanism of the trigger. So as you can see here, a hormone, it could be something like you exercised and you're trying to build muscle, so you're feeling sore, you broken down all the muscle fibers, now they need to be rebuilt. That's a classic example of how this, the mechanisms that are at play are basically addressing to the nucleus of those muscle cells, hey, we need more protein, we need more support around here, we're growing, we're exercising, and so that's how you get stronger and better. This is the one that we're going to talk a lot about today. This is the one that addresses cancer. This is the one that sits outside of the cell where the molecules come in, and here is the actual receptor itself. And what happens very simply is on a normal day, these molecules will sit on here, there will be a conformational change, and then these phosphorylation molecules will trigger these proteins, and off they go to do their business. What happens with cancer is that when these phosphorylation reactions occur, they come off, that's it, it's done. But somehow there's been a mutation which constantly keeps this on. It's like a faucet of a tap. You can't turn it off. You know, sometimes you have to change the rubber ring and you can't turn it off. That's what's happening. The rubber ring has gone and you can't turn it off, and it just keeps sending a molecule, a mechanism of action through here that triggers the DNA to keep churning out the protein, keeps telling the cell to keep dividing. I was working on it wasn't a disease, it was a family, normal family that had high bone mass. High bone mass is just a high deposit of bone, but it can be very bad for you. But this was a healthy one because these individuals, especially in the women for osteoporosis, they have really dense bones, 5x higher than normal individuals. And we identified the gene, and it was a gene called LRP5, which was in the Wnt pathway, which is also used by cancer. And the LRP5 molecule has 4 propellers. Think of it like a helicopter. But one of them, because of this very mild mutation, was twisted just enough to allow the signal mechanism to come in and stay bound longer, and so keep the signal open. And in a good way, this allowed those women who had that mutation, good mutation, to have denser bones. They didn't suffer from osteoporosis, and it took a lot of force to break any of those bones as well. So that's an example of a good mutation where the faucet's turned on, versus a bad mutation where the faucet's turned on and telling the cell, keep cranking, keep dividing, keep going, don't worry about why, but just keep doing it. And then after that, there's all sorts of other mechanisms that kick in. So again, to reiterate, but to delve deeper into where this molecular mechanism is driving towards, you can see very nicely that it is really a relay race where you start with the binding here, activating a protein from an inactive state to an active state that then passes that message down through this phosphorylation It's very simply just passing this phosphorylation. So you've heard of ATP, especially if you're working out. Well, you're going from ADP, so 2 phosphates, to ATP. And once that phosphate passed on, it goes back to its inactive state and it becomes now an ADP. There's also GTP to GDP and so forth. But you're basically taking a molecule that holds 3 phosphates that can then activate the protein. Then once when it's gone, it goes back to a 2 state, and that's it. It's very simple like that. But when that goes awry, then you have problems. And that's, again, alluding to how the cancer pathways take advantage of that. And then what happens is that the mechanism, it's really interesting. You'd think that it's just one should be enough to get down here, but it's just a chain reaction after a chain reaction after a chain reaction after a chain reaction to get yourself into the nucleus, to then sit on top of, and then recruit a whole bunch of proteins that would then sit on top of a sequence that they recognize that is all over the genome, that turns on a set of genes that then become players in what is being messaged through in and out. And then once when that's been made, the DNA makes RNA, it makes the protein, then those proteins go out and then they will do all sorts of things. And so here we then see another example of that, and I'm really trying to drill home this a little bit from different angles, how that mechanism of action comes in to the cell and how it drives towards the DNA to create a transcription and translation to make the protein that then responds to whatever the stimulus is. And in cancer, in most cases, it's creating new cells, creating new ways of bringing in nutrients. So you get angiogenesis, which is basically a whole blood system. So this whole tumor becomes literally almost like an organism within the tissue that it's growing in. And so it's got its own blood supply where all the nutrients are coming in. That's tapping off from the major arteries. And that is what Big Pharma's working at, trying to create drugs to stop that, to kill that, shrink that down, and then hopefully it will shrink the tumor because there's no nutrients coming in. So another example then of how these cellular responses are all about expressing genes. And so, as I mentioned, when something does go wrong, then you get this division occurring. Lots of cells start to divide, grow, and mutations start to get placed in there, which are not good mutations. These are driver mutations as opposed to what I call passenger mutations. And so these driver mutations are driving the cell to live and turn into something which is not good for anyone to have around. And so you can see very simply how these growth factors activate cell division. Cell division then is really the molecular basis of how the cell can then grow into another cell and another cell, and when that's out of control, then you have a problem. And that's what you folks are going to basically pick up on. You're going to basically say, I know what the mutations are in these cells that are affected with, that are related to a tumor or a cancer, and from that the physician's going to be able to make a really good assessment for the patient as to what therapeutic drug is available for them to try to get to these cells and shut them down. This is really how we've evolved. Within here then you have this nucleus. It too has like its an envelope, a barrier, and there are pores so that things can go in and out. So RNA comes out of here, proteins from the messaging that I told you about go in and they attach. You have a nucleolus, it's almost like a nuclear reactor going on in there. And so this in itself, as written down here, is a command center. It dictates basically how the cell will behave, from living normally to basically saying, all right, guys, time's up. We have to now go into what's called apoptosis, which is basically killing itself and moving on because it's done its job, to basically when it goes awry, things go wrong, where it just says, right, we just need to keep turning this machine over at a higher pace and creating more and more cells of a type that are not supposed to be there. And that's where the genetic material has gone wrong. I love this picture turning around here, because basically what you have here is a double helix, and you've got this phosphate backbone. This piece of DNA revolving around here really shows you the bases, how they're stacked on top of each other. And in this cartoon here, that you can see that what comes off is a base pairing of a type that is held together by weak hydrogen bonds. Those hydrogen bonds are like a zip on your jacket or on your fly, and it just literally unfastens. The energy required is not that amazing to break, and it just comes apart, and then it gets used to either copy or transcribe pieces of it so that it can make RNA, or, as I said, when it's going through mitosis, which we'll talk about, for cell division. So zooming in a little bit and getting some connections around these bases and how they connect, there are some rules. A always goes with T, and C always goes with G. And if you look at the AT connection, there are 2 hydrogen bonds. That's a weak connection. And in fact, we'll talk a little bit about PCR and how, when you're actually creating primers for amplification of DNA, that these rules of this 2 hydrogen versus 3 hydrogen in the GC really impacts the thermodynamics of how PCR works. A goes to T, and G always connects with C. However, there is a slight change in the rules when it comes to converting your DNA to RNA. RNA has, instead of a T, it has a uracil in its place, and just slightly different in its structure right here. And RNA is kept as a single strand. And the idea here is that once when the RNA is made, it then gets shuttled off, and then a whole bunch of proteins will come in, and they need to read that RNA, and they need to have it as a single strand to be able to then read these triplets that become an amino acid, and I'll talk a bit about that. And these amino acids come together to make the protein. So RNA is always single-stranded, and it always has a uracil in place of the thymine. You go from DNA to heteronuclear RNA to messenger RNA, which is all the introns have taken out. It's now collapsed down to one big open reading frame, and then translation occurs, and then you get your protein. So let's start and talk about transcription, where we move from DNA to RNA. You saw how the DNA opens up, okay? It opens up, and then it copies everything on the bottom strand so that it gets a perfect, mirror image of that. The top strand is not used. Unless it's in replication, then the top strand, as you saw, gets spooled out, copied, spooled out and copied, whereas the other one, the polymerase just charges along and copies it. So that's for cell division. For here, it uses the bottom strand where it doesn't have to spool it out, copy it and spool it out. It uses the bottom, and it just literally opens up the DNA, and the polymerase sits onto the promoter, an area that it recognizes at the beginning, and just charges straight down at thousands of nucleotides per second, which is almost equivalent to what the ion torrent's doing as well. So the RNA strands created based on the DNA template via the complementary base pairing, and then the genetic code transfers from that over to the RNA, and then it's ready to move to the next level. Now, as I said, after the intronic sequence has been removed and you now have what's called a messenger RNA, the next step comes in where it starts to read. The ribosomes come together and it starts to read what this code is all about. And 3 bases make up 1 amino acid. And so by going along and reading, it's like reading a book. You just read the words and it just brings the pieces in. It's almost like an SOP, standard operating procedure. You just follow the instructions, and if you follow the instructions, nothing can go wrong. Sometimes things do go wrong, but in general, look where you're at in your lives. If you're doing good, you're doing good because things are going right. So it's doing a great job. And this mechanism occurs thousands and thousands of times per day, from the very beginning to the very end. And all it does is it recruits these, what's called transfer RNA, which has attached to it 1 amino acid. And by reading the triplet down here, this transfer RNA sits down, and then boom, it bonds the 1 amino acid to the next one. So it makes this long strand of amino acids. Now, again, I'm not going to go into horrendous details about post-translational modification, but simply put, once when this single strand is made of amino acids, it's folded up to become the functional protein, and it has certain attachments to it that drive how it functions within the cell. But this is very much simply how it all gets put together within the ribosome with the transfer RNA molecules bringing in the amino acids, and then as you can see, once when it's connected, it then shuffles out and picks up another one and ready to come back into the process. And here's, if you're interested, how people look at determining what that messaging is all about, what that code means. And so, if you recall, I mentioned that RNA has U's in it instead of T's. So what we have here for this decoder is, instead of a T, there's a U. And each one of these triplets codes for different amino acids. But note how in some situations there appears to be 4 types of triplets that code for one amino acid. Superb redundancy. You've got to have redundancy in the system. If you don't have redundancy in the system, if something goes wrong, then you could be in trouble. But let me explain how these come together. We'll use this leucine as an example. If you go from here, C is the first base, to U is the second base, to the third base over here. So that would translate from the DNA perspective, CTT. Now, in the world of DNA, there's what's called a third base wobble, and so in this situation you can have any one of these bases sit in here, and because of that wobble you are allowed to it will still recognize it and put a leucine in there. Now in some of the more stringent ones, you've got here tryptophan, which is a UGG. See how close it is to a UGA? If there is an A there because of a mutation, you're going to have a premature protein. It just stops dead. So it hasn't completed the full length, it's got half of it, and usually it's not functional, and it gets taken away and destroyed. Now, it could well be that there could be some functionality to it as well, depending on where in the process it occurred. So that's something to think about. And note as well that there are 3 types of stops. So third base wobble is good if it's within the group that's allowing for it. Not so good if it's outside something as unique as this. And there are 3 types of stops. And then there's a start. Like everything in life, we've got to know where the beginning is and where the end is. And so every amino acid has a methionine in it, and that's AUG. But note how close it is to isoleucine. So again, if that happens to be at the beginning, or it can't find something similar, then it might look somewhere further down to find the AUG. It will not start unless there's an AUG.
Question, Richard. So, if I'm following this correctly, DNA splits in half. Half that essentially RNA drops to T. Is that correct?
Well, it doesn't drop the T. It just replaces, when it's making the RNA, it just pulls in the uracil instead of the thymine, because it's a different structure to what DNA is, the RNA.
The RNA is essentially messenger, and somebody else pulls in amino and turns that all into a protein.
Correct.
The way you've described that's perfect. And so, looking at this chart then, you can see, and you can actually start to create changes yourself. You can actually insert mutations. People do that. They take pieces, and I've done that myself. You take pieces of DNA and then you insert mutations in based on the open reading frame. You want to see the impact of changing an amino acid, or you want to try to replicate a mutation that's reflective or that's found in somebody who has a particular disease, a Mendelian disorder, single point mutation, and you're looking to work in a dish of sorts, looking at cells, the impact of that. So you can go and create and change things. But this table is really powerful because it gives you a good sense as to what the amino acids are. So with the 4 codons, A, G, C, and T, makes 20 amino acids. And those 20 amino acids are mixed up in all sorts of combinations. And these 4 codons are also mixed up in all sorts of variety of ways to give you basically 25,000 genes Now, the hubristic amongst us many decades ago thought we actually had 100,000 because we're humans and we're very complex. But when the genome finally got sequenced, it was found that it was 25,000 genes, much to the horror. And then it's like, well, how can it encode for all these proteins? It basically encodes 1 million proteins. And that's because there are subtle differences. If you recall, when you take out the intronic sequence, there's also an opportunity to take out an exon, and that's called alternatively transcribed products, which will give you the same protein but with a slightly different twist. And that slightly different twist is very specific for cell types. So that gives you then your million cells plus the fact, million proteins, plus the fact that, as I mentioned, the post-translational modification, those two add a different layer of complexity to the protein in the cell type for its function. So that's where the million has come from. But yeah, it's only 25,000 genes, so sorry folks. You're pretty close to bacteria. There were about 4,000, so but it doesn't matter because there's so much variation that can be had that you get the complexity that we all are. Point mutations. Okay, there are a variety of point mutations that are out there. A point mutation will cause a change in the protein structure. It could be good or bad. So a missense mutation will cause a different amino acid to be used in the protein, as I mentioned here. A nonsense one is one where it causes the end of the translation and the premature termination of the protein, which I mentioned to you too. And then the final one, which is always a good one, is a silent one, where the point mutation creates a codon that codes for the same amino acid as the original. So that wobble I was talking to you about. So those are the key major mutations that are out there that can cause either a start to disappear and it'll find it further down and create a protein that has no relevance whatsoever, a stop to disappear or to be introduced, and other amino acids where you've taken one out and put a new one in. And in that situation where I told you about with the family with the high bone mass, they had the same thing. It was a mild mutation, but it turned out to be very, very much an evolutionarily good one for them. And then here's an example then of a mutational change that occurs in sickle cell anemia. Now, sickle cell anemia is interesting because on one level it's not good for you, but on the other level, depending on what environment you're in, it's actually very good for you because it doesn't seem to have a problem if you're living in a society or an environment where you've got mosquitoes going around with malaria. It's a really protective thing, but sickle cell anemia doesn't really bode well if you're not in that environment, and as you can see, you get anemia. So it's an interesting mutation that's evolved, and here it is. It's the wild type at this juncture is CTT, and the mutation has changed here in the middle. So it's gone from a CAT and replaced the adenine to the uracil, which was what comes up on the message, but the DNA is reading as a T, a thymine, and then you just get this glutamine to valine change. And like I said, you end up with sort of anemia, but yet you're protected against malaria. An interesting evolution that's occurred there for...
Is wild type just normal? Is that another word for it?
Yeah. So you have a genome that's wild type.
Well, how did it get wild type? Normal
that's
wild
from
type?
genetics way back in the 1800s that refers to wild type and mutation. So I always look at this. It's kind of an interesting, there's an interesting idea that's out there which says let's sequence the baby when it's born. That's your genome that you're born with, if you like, wild type, right? And so, but as you know, as we grow and the environments we're in, we're going to end up with what is called post-zygotic mutations. These are mutations that occur in us because of the way we eat, where we're living, maybe it's in an industrial environment, and so the impact of the environment will cause mutations to occur. Maybe you're sunbathing. Things like that cause damage to the DNA that for specific tissues is very relevant where that mutation's occurred, and that's how, again, cancers occur. So those post-zygotic mutations are what drive you to become basically diseased or not diseased. and how some of those mutations occur and they're meaningless, and others become actually life-changing. Okay, frame shift. This is a fun one to explain as well, because basically what's happened here is that either in this case, a G has been removed, and now it's not reading methionine, glutamine, alanine, and glycine. It's shifted it so that this AAG is recognizing as the next triplet. Okay, remember, it doesn't go in and fix things up anymore at this level. There are mechanisms during replication that go in and double check. That's why you have two strands, so you can look at both and say, whoa, there's something not right here. What do I do? Do I put one in? Do I take one out because that one's showing that the template should read this? So in this situation, something got knocked out, and that was the G, and it shifted everything by one. And so you can have a situation where a base is actually added in, or in this case deleted, and then it alters the protein. Now sometimes you can get real lucky and it can actually go out of frame, if you like, and then back into frame, and you may end up with 2-3 amino acids that are out of whack that may not be too crucial in the folding of the protein, and it's still functional, and it may impact you in a mild way. So something to think about. The complexities are amazing. And then if you look at this great little line here, the red fox ate the fat rat is the wild type sequence. Then if you have a point mutation where it's changed it, called a substitution, it still reads the fox ate the fat rat, but now it's the ked fox. So now it's like, all right, can I still function? And then you have a frame shift where you have an insertion which shifts everything off, and now that triplet structure has gone completely awry. There is still there, but now you've got Cray, Defoe, Zat, Ethi, Effer, Tra, and so forth. Okay, so that's the two things that happen out there. This one might slide in, actually, and get away with it. Again, depends on the function, and if you do have that, it could be enough to create the mutation that could be life-changing for you too. All right, moving to human chromosomes. Let's go up the scale. Let's get to the big boys and girls that sit out there and encode all these amino acids. These are the 22 chromosomes and the 2 sex chromosomes, X and Y. You can see they're all laid out based on size, and that's how they originally used to do it. And then they have this staining called Giemsa staining, and it stains black and sort of a low color, and that basically refers to the density of the genes, where the genes are. So in the black areas it's more genes, in the white areas there's less genes. To put it simply, AT rich meant, before all the sequencing came out, less genes. GC rich meant there was a lot of genes, because GC areas were areas that suggested there were a ton of genes, and those were areas that had promoters and all sorts of coding areas that brought proteins in to get the genes transcribed to make the proteins for you. So that's how it was originally looked at, and people still do this. It's done more automated. I've done this myself. It's really hard to do. 46 chromosomes, that means that there's one of each that comes from your parents, and we'll talk about how those come together to make the 23 pairs, and each chromosome contains DNA that encodes all the genes. So this is a pretty picture, and when we go through later on, I have taken pictures of these chromosomes to denote where many of the genes that are up here sit, so that not only do you actually get to know a location of like 1q23, but you actually know where 1q23 is and where that gene is. Again, just to give you some tools of knowledge and understanding, just to really help you out there so that you don't feel, I never understood what that meant. Okay, so even chromosomes have mutations. They are giant ones, so let's go through them. They're not too difficult. So you have amplifications, where basically you have a whole chunk that's just been copied. So you've heard the term aneuploidy. That's going to be multiple parts of the same chromosome. So Down syndrome is an aneuploidy where you have 3 chromosomes. So in this case, you may not necessarily have a full chromosome duplicated, but you may actually have just a piece of it duplicated. Literally just all got screwed up in the system. You then have deletions, where you can see here a huge chunk has just been disappeared, just came out. And now you've got genes that are gone, and while you may have another chromosome that's perfect, if there's a need for 2 copies, or again based on the expression, it needs a lot more of it, you could find yourself a little short there. So while 2 chromosomes are good to have, sometimes it's not always that good, and depends on the genes that were in the section that were out. Then you've got these juxtaposing mutations. That's bringing together separate genes to form functionally distinct fusion genes. So that basically means that a chunk went out and it took half of one gene, half of another, brought them together, and now you have a new gene. It's a fusion. So you got half of one, half of another, and there's an example I'll show you of that, and how tests are done to detect that. I think one of them is for lung cancer, EML4 and ALK, and they run literally, one is encoded on one strand and the other one's encoded on the other strand, and then this chunk's whipped out and they're brought together. It's kind of flipped, it's whipped out, and then you make these genes that weren't supposed to be brought together, and that with a defunct protein can give you cancer. So that's there's tests out there as well to detect that. So chromosomal translocations is one of these things, where the interchange of genetic parts of non-homologous chromosomes. So literally it's pulled one from, it's taken this piece and connected it to this piece, so you get a new chromosome or a derivative. Interstitial deletions, where again you've got the same thing going on, big chunk moved into another piece of the chromosome and makes it bigger, and the other one now has become shrunk. And inversions, where it's just taken a chunk and just twisted it upside down. And then loss of heterozygosity is one of the key paradigms of cancer, where you get a multiple hit. So if you've got loss of heterozygosity, you've got one's disappeared, you've got now one gene active, that's it. Right? It's like having 2 kidneys. You know, people give one up, kind of give one up to another friend of theirs, but if something should ever go wrong with that kidney on the other person, it's going to be, they'll be in the same boat. So you have one gene. The reason why you get cancer is because this one's disappeared, and then the mutations come in, bang, it's upset that that gene, and that gene now is basically the one that's driving your cancer. So loss of heterozygosity is very important in the paradigm of cancer. Okay, and then, as you saw from that great movie, how it all folds up. I love that movie. You got your DNA. It comes together with these histone proteins that just slot in, and then it's just amazing how you can take 6 meters of string and coil it up into something so small. And then it goes into these nucleosomes, then those nucleosomes are bundled around into coils, and then those coils are bundled around into supercoils, and then those supercoils are bundled around again to make what's called sister chromatids. So this is one chromatid, this is a second one, anchored together at the centromere. This is the classic picture that you see in textbooks. These are sister chromatids, they're identical, and this is the chromosome, and the ends are the telomeres. Now, you probably heard some stuff about telomeres, that they encode your longevity, and clearly it's about, it's really, at the end here, it's almost like every time a cell divides, a piece comes off, and so the cell recognizes when to say it's enough, we're done, and that's either old age or the cell has to collapse down and go into apoptosis. But clearly telomeres are something that people want to try to address when it comes to living longer. Try to stop those telomeres from shortening. But it's not always about short telomeres or long telomeres. It's about how those telomeres are shortened during the process. And of course, lifestyle has a lot to do with it. If you're a relaxed, laid-back person in an environment, a monk maybe, where you have nothing to worry about in life, then maybe it doesn't matter if you have long or short, you're going to live for a long time, versus those that live in the 21st century in America and trying to make a ton of money, and you know, well, all the things that will grind you down, that can impact how those telomeres are addressed, so stress, environment, etc.
Richard, do we know how long it takes for a gene to mutate environmentally? So I'm living somewhere in particular, I moved from the islands to the U.S. or from China where life is different to the U.S. And how long does it take for the gene to kind of shift and start doing the crazy stuff?
Well, again, I think when you talk about shifting, how long it takes for a gene to sort of, I think the question maybe, if I may be so bold, is how, if there is a change, no matter whether you moved from the islands to a more aggressive lifestyle, it's how long will it take for the impact of a mutation to occur if one has occurred? And it could happen on the islands, by the way. It really depends on the impact of the mutation and what it's driving towards. If you notice, cancer really is an old age disease. We get it from about 60 onwards, right? And I'm very familiar with lung cancer, and it's such a shame with lung cancer that you could smoke and the stats are on your side. 85% of people who smoke do not get lung cancer. But those people that do, from the age of 60 upwards, get it, okay? It's the people that never smoke that fall into this category. And there's 90% of those are the smokers, 10% of these non-smokers. They happen to be mainly women and at the age of 40, and there's pretty much little you can do. The head's shaking here. Somebody knows about this one very well. If a mutation occurs that is very detrimental, it can occur pretty quickly. If it's one of those slow growers, something that just happens. You know, a mutation, by the way, we all get mutations within us, and we all get cancer cells within us, but they get taken away. They didn't make it. There wasn't a driving force to push that cell to become the next one. And I have a picture in the next section that shows you how you sometimes, while the model of one hit, that's it, you're done, it doesn't always go that way. It needs more hits. And if you're going to do like sunbathing, if you insist on sunbathing and getting a cool tan, then, you know, it's going to be several mutations that are going to drive a melanoma. So it really depends on the mutation, and it really depends on how it's going to impact the cell, and whether your natural system can get rid of it, and it's done. It just didn't make it for whatever reason. There is no one's measured it, but clearly, as you've read, lifestyle has a heck of a play on how you can prevent things from driving towards that. So stick to the islands, best place. Okay, let's talk about division. There's two types of division. There's mitosis and there's meiosis. I'm not going to go through all the processes and the names because that's a course in itself. The books back there will help you. So again, as I say, do feel free. But there's two types. Mitosis is basically all the cells in our body, they're just cranking out. They're dividing every day, and they have to copy each other to make another daughter cell so they can continue doing its processes. Meiosis is where, upon fertilization from the sperm and the ovum that come together, bring in 23 chromosomes to 46, you get a recombination event. So once when the recombination events occurred, everything's all mixed up. Then you go into mitosis, which is the cell division, which then is programmed to become different tissue types, etc. So briefly, if we talk about mitosis, which occurs in our somatic cells, as put here, it's the bulk of the body. The cells, they all line up, and they literally just peel away from each other, and you saw from the movie how it's copied. They just split and copy, and then as the cell's made, you get a perfect 100% reproduction. That's the aim in the daughter cells. So for meiosis then, this is where, as I mentioned, you have the 23 chromosomes from the two parents come together to make the full 46, and you get that recombination event occurring. That's where the sort of mixture occurs, and then after that, everything's done. Then it just basically replicates like crazy, and then there's all these mechanisms that we've talked about that come into saying, right, you cells become sort of the brain, the kidneys, and so forth. It's a little bit more complicated than that. And then finally, let's talk about a gene. A gene's a gene. It's, as written down here, it's just a segment of DNA. These are stored in chromosomes. They're passed down. These are what we see, good traits, bad traits. They're all the ones that drive the phenotype that we see in our children, and that they pass on to their next generation as well. Some things are kept, very strong genes. We'll talk about those dominant genes, and some become recessive and they get lost, like my children, as I said, have blue eyes. I have the brown eyes. My father has brown eyes, but my mother has kind of blue eyes, so maybe I'm heterozygous, which means I have brown and blue. My wife has sort of bluish eyes, so both my kids may have got the two blues, and it was more powerful, those two coming together, than the brown. But if I have more kids, not that I want any more kids than two, but if I have more, maybe some brown may have come through. So, but the blue's working, and so the brown's gone. The nose is gone, the brown's gone, the colour, the hair's there, but anyway, okay, let's talk about alleles. You've heard this term alleles. Don't be afraid of the term alleles. You will hear people talk about them. They're just basically two components within a gene that have differences to them. They can either be the same or they can be different. And if they're the same, you hear the word homozygous. If they're different, they're heterozygous. So walking through this then, you can see that in this case here, the copies are not identical. And then because of that, they're called alleles. They're all alleles, actually. But so, as I mentioned, if these alleles are the same, then they're homozygous. If they're different, then they're heterozygous. And that's where you get that mixture coming in. And then you can look at this pattern here and come up with what's called a genotype. And if we just use black and white, then you can see how this genotype will be WBB, and here is BBBB. Yeah. So that's how you form the genotype, and many a brave soul has gone in there to try to genotype a whole individual, and the complexity behind that is immense. Just looking at one gene that's associated with the disease and looking at all the variants that drive some of the phenotypes is very, very complex, but that's what an allele is. And then, as we talked about, dominant and recessive. This is what we're going to talk about for the next 5, 10, 15 minutes, and how that plays a role in disease and how it plays a role in cancer. So, basically, think this. Dominant dominates, recessive doesn't. Unless two recessives get together and gang up on the dominant, then the dominant loses. Okay? So two dominants against one recessive, forget about it. But two recessives against one dominant, you're in good shape. So that's how I would look at it, very simply put. And so you can see then for these blue eyes, which is a recessive, and the brown is dominant, then you can see that this person here is heterozygous. This person here is homozygous, and then this person, which is recessive but is homozygous for the blue, and so in this case here the brown wins, but the blue wins here, and of course naturally the brown wins there. So as I mentioned, if the allele is dominant, then it's going to be the one that's expressed, and the recessive is masked. Okay, and then if the individual receives the dominant allele from one parent and a recessive, then the dominant will always win. Unless you get, in this case here, two recessives come together to take on the dominant, it's bye-bye dominant. And here we see a great example here with our little friends, the mice, where it's albinism, loss of pigment in skin, hair and eyes. Also seen in humans as well. And again, these two little mice here, they have a genotype. So this guy here is big P, big P. His phenotype is normal. He's as good as he can be, so no problems there. And the big P is the dominant allele. So in this case here, with this mouse who has the big P and the little p, he's going to come out normal. He's in good shape. Now this little guy here, he's got the two little p's that come out, so he's got it all. So he's got no protection. And we'll see further down in this presentation that this person is carrying this little p here, and if he mates with another one that has a little p, that's how this little guy comes out. And think of cystic fibrosis. It's a classic example of the fact that 1 in 20 people actually have this type of combination, the dominant, no cystic fibrosis, and the recessive. And then if you mate with a person who has the same, then you're going to end up potentially with the little p, or in this case, with the disease. So cystic fibrosis is quite a common one, as you know much about, and so that's how it all comes together. And I think, you know, one of the things that I've read about as well is in sperm banks that all of a sudden they look at the picture, the guy's a genius and he looks like he's Nobel laureate, right? The whole works, boom, the child is born and has got cystic fibrosis. One, of course, they don't check for those type of things, and two, obviously the mother wasn't checking herself too. So, and that can happen. All right, this is called a Punnett table, folks, that, again, might come up in your conversations, or at least in the sense of how these traits are pulled together. And again, very simply, you have the big P and the little p on one side, big P and little p on the other, and you can see then in that example of how you get homozygotes and heterozygotes, you can see that the big P dominants come together to make that mouse. You've got the heterozygotes, the two heterozygotes, and then the little p. So this is all from Mendel's genetics with his p's and all that stuff, although some people have said that he cheated a little bit. Irrespective, he rounded up the numbers or rounded them down. The bottom line is that you get more heterozygotes than you would homozygotes. Okay, human Mendelian diseases, these are the easy ones, if you like, in the context of there's a single gene disorder. And those are the ones that have all been identified. They're rare, and they're called single gene disorders, rare orphan diseases, or monogenic diseases. It's basically a very simple situation of the dominant gene having a mutation in it which is expressed, and you're expressing basically the genetic mistake. And so you can have them as an autosomal dominant, autosomal recessive, X-linked dominant, and Y-linked. Autosomal, it's the chromosomes that are 1 to 22. The X-linked, of course, the X and Y chromosomes. And then, of course, you get all sorts of mutations that can drive any of these diseases that come up. And that book down there's a couple of books that describe every single genetic disease out there. So if you know somebody's got something, McCusick's book, it's also online as well. But it's always a good little thing to pull off the shelf and just say, oh, I remember somebody mentioning that and knowing what it is. Again, same types of mutations occur. Insertions, deletions, copy number variants. So the insertion can be a single base, it can be a triplet. Fragile X syndrome, I worked on that one. That came out to be a triplet, which is kind of interesting. So triplets, that can, expanding repeats can be a problem. Huntington's, I believe, is an expanding repeat too. And deletions, again, something gets knocked out. And copy number variants where you've got too many of the same thing. Oh, and this one's very interesting. I'm not going to go into too much of penetrance. When you read about it and you hear them talking about it, it basically means that the dominant gene has 100%. It's not going to give you any variation of less than or stronger than. It is what it is. Bang, 100%. Now, that's the rule. Does change a little bit, and it can change the calculations. But when you look at complex diseases like asthma, diabetes, and all that stuff, where there are many genes involved, that penetrance is no longer 100%. There's too many genes. They're all playing their role. So that's why you get this broad phenotype of, take an asthma person, mild, moderate to severe, because the penetrance of all the genes and the way they're acting on top of each other is variable. In one gene, one mutation, it's 100%. You get it, you deliver on it. As a phenotype, you express it. So I thought we'd run through this as a quick example of how pedigrees look and how they come together. In an autosomal dominant, remember, dominant expresses. In this situation here, anybody who's got a bit of a blue in it is affected. However, if they've got a bit of white in it, they are heterozygous. They have both the dominant gene and the recessive gene. And if they're all white, they've got nothing. They've got the good genes. And if they are all blue, then they are homozygous dominant for the disease. So remember, 2 chromosomes, 2 mutations, copied through, so they pass it on. And you'll see here quite nicely then who gets who. So in this autosomal dominant, it's one of any disease within chromosomes 1 to 22, you can see that you can get an example of one chromosome going over to this lad and one from his mother. This one here, the girl here, got one from the father and one from the mother, so she's good. He's affected and also has the gene, naturally. But if he ever decides to, as in this case, get married, then the progeny can be very much normal because of the normal gene, because he's heterozygous. Over here, you've got two heterozygotes, both affected. And in this example, the young lady here got lucky. Both chromosomes that were the good ones, the normal ones, came through. So she's not going to pass it on, but she married this guy who's affected, and so her progeny are showing up. But note, less and less are coming through. So it's diluting out. The gene pool is getting rid of it. But in this situation here, where they're both heterozygous and affected, there are more affecteds coming through who are homozygous. So making the situation even more complicated there, and you're not diluting out the gene pool of this disease. And you can see that some of the diseases down here, the frequency, many of these rare diseases can range from as high as 1 in 500, which is not that high, to 1 in 100,000, 200,000. In this case, we have recessive. Remember, recessive, you have you can have the gene, but you're not affected. So here, if you have a color, you're not affected, but you are carrying. Okay, so these two blues, this is the classic cystic fibrosis. They're carrying, they're not expressing, but unfortunately the girl here got both the chromosomes, so she's now a homozygous recessive, and she's expressing the disease. These two are carriers. Here we have somebody who's homozygous recessive, expressing the disease, and you can see how the chances go up even more, and so forth. And so you can see some of the diseases down here, the ranges and the frequencies. Here then you have, from an X-linked, it's going to be X and Y and XX. Now here the father's expressing with his X, and he will pass his X on to every one of his female progeny. The X's from the boys always come from the mother. The Y comes from the father, no doubt. So in this case then, the female picks up the X from the father. It's a 1 in 2 chance will express. And the same over here, where the mother, who's going to pass one of her X chromosomes to every one of her progeny, the boys will express. And then in this situation, again, it's this. And this is a classic sort of example of a disease that I worked on with X-linked adrenoleukodystrophy. Pretty sad situation of an X chromosome that sits in the mother, which is mutated. It's XQ28 right at the tip of the telomere. And the boys are born, they look normal, and then by about age of 3, things start to go wrong. And they start to very much waste away. So by the time anywhere between 8 and 12, they're like just skin and bones, and they're very much mentally challenged, and they just end up dying very young. And so something you can't do nowadays, but I was doing my PhD, and we designed a test to help determine whether the fetus was carrying the X chromosome with the disease on. And this was part of the National Health Service in England, up in Birmingham. And I published a paper on it, but clearly it worked and was very accurate in determining whether that fetus had the X chromosome and was going to be a challenge to the family. And it's a very tough challenge, especially families that are somewhat underprivileged as well, to manage a child like that. X-linked recessive. This is a classic case then of where you have the protection carrier, but again it's recessive. And again here, because the X-linked is coming from the mother, the boys only get 1 X's, and that 1 X is from the mother, so she's recessive. Sorry, she's homozygous, so they're going to be affected. And so these two here are not affected. They got one from there. They actually should be this. Anyway, okay, and then the last one, which is a very simple one, Y-linked, it comes from the father. So you can see all the boys will get it and nobody else. If the father is affected or not affected, then the boys and girls are good. Well, boys, I mean. Okay, complex disorders, as I had mentioned, and we won't be doing any of those type of diagrams because they're just too complex to follow through because there's so many genes involved. Basically known as multifactorial or polygenic. These are human genetic diseases that have multiple causative mutations, and as I mentioned, it explains the broad phenotypic expression that you get from mild to severe. And you know, somebody who's got asthma on the one end of the spectrum and then the other one saying, well, what's the difference between you and me? It's all about those genes, where the mutations are, and this is where I was talking earlier about the regulatory mutations, the ones within the introns. This will become something over the coming years to be a very big discussion point, and identifying those and understanding how they play in these complex disorders. And by the way, cancer is a complex disorder. It's not one gene, one mutation. There are multiple genes, multiple mutations, and they all, as you'll see from the pictures, play a role in this incredible complex wiring mechanism. As I mentioned, it's not clear how things are inherited. It's really difficult to manage as a disease because of so many genes involved. If you have one gene, you know you can direct your drug towards that. And with cancers, it's not always a case of here's the cancer, here's the mutation, here's the drug. If a cancer has multiple mutations within it, then some of those mutations will be in cells where there's not that many cells with it. So you end up with a situation of getting rid of the majority of the cancer, and the person goes into remission, but then you end up with a signature of a relapse signature. The question becomes, what do you do with that information? And is there a therapeutic treatment out there to deal with that signature that you've now just identified? So these are some of the sort of legal issues that are coming out with all this information and how you manage it. But the complexity of these disorders is really tough. Even something, as I keep mentioning, asthma, it's a tough one to manage. What's the other one? Autism is one on here. It's another one, right? You look at the phenotype and you go, really? But again, it's a broad range. ADHD, I mean, that expresses itself in so many different ways and so forth. So there's just so many of these genes that are at work here. And as I mentioned, the penetrance in bold, not 100%, okay? So, because it's just difficult to monitor. And there's some of the good players down here. As I mentioned, the cancer is one of them. Okay, predisposition to diseases, we're nearly at the end of this section here, really is about what you're born with and will you express that. So it really becomes, from a complex standpoint, protective versus at-risk genotype. Okay, so if you have an at-risk genotype, you could express severely to mild. If you've got that protective one, even though you have some of the at-risk alleles that we talked about in there, if there's enough protective in there, it could override it. You know, we all get sort of like, you go out and you get sort of runny nose and that, and you think, oh man, it's that season again, right? But that's it, you're done. Maybe something, just a day. So you've got a lot of protection going on there versus somebody who just can't even be out at this time of the year. So that's what that is. And the environment is a big, big player in this. Genetics is 50%, environment's the other 50%, especially in complex diseases. But if you are predisposed to a disease, you will get it. Okay, and that's in one of these ones where 95% certainty we know about breast cancer, BRCA1 and 2. If you have that mutation in that gene, the likelihood is 95%. Huntington is another one where there were some big issues when this was found out because the onset is until 40. So the question now becomes, do you want to know, right? Cystic fibrosis, again, knowing early is a good one, and Fragile X syndrome with that repeat that's been identified is a good one. Again, so you have a child that's born, looks normal, then all of a sudden starts to show some symptoms, and then in the end it's like, well, let's check the genome, and it's Fragile X syndrome. So predisposition, it's going to happen. It's in there. You're born with it. What happens during your lifetime that changes that is what leads to cancers, okay? These are new mutations that have occurred. These are ones, these predispositions. And I'm a big believer of predisposition as well, because you could well be born with mutations which, if you're in the right environment, may actually put you in a susceptible situation to getting that disease. So environment and the genome, they do work together, no matter what people say. So the penetrance, I really just wanted to show this slide. I found this picture, I thought it was really cool because it really describes to you what it is you're up against when you're dealing with complex diseases and a simple single gene. You can see the penetrance is high for a Mendelian disease, okay, but hard to identify genetically. It could be anywhere down here. These are very rare, of course, and then when you get to the most common diseases, it's if you've got penetrance, it's not going to be high. It's rare. It's going to be very low. You just don't know. In genome-wide association studies, these guys have gone in and they've searched genomes in thousands of people for various diseases up and down the wazoo, looking for mutations that drive the key drivers, if you like, to those diseases for these complex syndromes. And they haven't found anything that stood out. And there's a whole load of reasons behind that. But that's because part of it is penetrance. And then finally, genetics versus genomics. Very important to understand this so that you don't sort of come off like, does that person know what they're talking about? Genetics is really the BRCA1, the BRCA2, Huntington's. It's the gene. One gene, some mutations. Genomics is that complex ones, okay, where you've got a whole load of players all working together to create the phenotype at varying levels. So that's studying multiple genes, their expression profile as seen here in this multiple, in this array, the interactions, how they interact with each other in the pathways, whether it's some are really interacting, exerting that mutation more than others, the effect of mutation, the modulations, how it's all modulated, and the environment, of course, in all of that. So this is a lot more complex. Cancer is part of that too, and then genetics is just the single gene. This is the foundation to the iron torrent. There's history here, and you know the guy that developed this, Jonathan Rothberg, he's a smart guy. Although Fred Sanger probably takes the biscuit on this one because I think this chemistry is amazing. Really simply put, as you know, the bases come together, as I showed you, but what this does, very simply, is that Sanger basically threw into the cocktail this dideoxy. And very simply, it has a hydrogen in place here instead of the hydroxyl. So when a base comes together, you've got the 3 phosphates, 2 come off, it, boom, joins on. But if there's a hydrogen ion here, it just stops the termination, dideoxy sequencing. And very simply, that's it. And you have a cocktail of your nucleotides with this dideoxy, and you end up with this. You end up with this old-fashioned ladder of beautiful sequence, which I used to do myself, and you would read it from the bottom all the way up, depending on how you loaded your gel. And you would be able to read, very simply, A, T, G, C, all the way up, based on what terminated here. And because there's a lot of molecules in the reaction, you get a good, strong signal. Oops, sorry. You get a good strong signal and you're able to read it off the autorad. Now, trouble is, these are called autorads, autoradiographs. Trouble is that sometimes these autoradiographs will smile like this, and so you'd have to try and read your sequence. But you would be able to read it quite nicely and look for mutations, insertions, deletions, whatever you want. Beautiful, easy. It was with radioactivity in those days. This guy here, with his colleague Maxim Gilbert, both of these guys got Nobel Prizes, by the way. This one's a chemical reaction, a little bit more complicated. I've tried it once and didn't like it, but it's got noxious chemicals, all sorts of stuff. But it allows you to do very much the same thing, and you can read the ladder all the way up. Very nice, very simple, very eloquent. And then it got a little bit more complicated. When ABI said, well, why don't you put some colors on it? And if you put some colors on it and you put it in a machine, and the machine has a sensor that detects the colors, you can actually read a lot more, and you would end up with 96 samples on a gel, and the sensor would literally read as it came through, and you'd be able to see on the computer this. And then when it was finished, you got this nice chromatogram, and everything's on it. It's read for you, and then you would have a file that you can cut and paste, blast it, and you know what you've got. Very simple, very eloquent, and it's using the same chemistry as Sanger. And then you can get into doing all sorts of things. You can get into making genomic libraries to sequence, cDNA libraries. cDNA libraries are basically all those message RNAs that I talked to you about. They actually, you collect all the RNA and you convert it back to DNA, and then you are able to clone it and sequence it. And that's how people understood what cells were expressing, genes. Genomic libraries for the sequencing of the genome in 2000 and the completion in 2003 was very much simply taking the big chunks and just cutting them, all sorts of things, ways of cutting it. Plasmids would take anything up to 10 kilobases of genomic DNA, cosmids up to 40 kb, BACs and YACs. I've used all of these in sequencing and trying to find genes. Crazy days, fun days, exciting, and I was working in a company called Genome Therapeutics that sequenced chromosome 10. It was the only publicly traded company that was part of the human genome sequencing, the public one. The other one was Craig Venter, who did it privately. So he did it alongside with the publicly funded one, which cost $2 billion. Anyway, so these were the vectors that we used, and there's lots of information around them. I just want to give you some history there about how sequencing was used, because this is really what's happening now. Genomic DNA is being sequenced. Never mind the cDNAs anymore. People do that, but not so much. It's all in the genomic DNA libraries. And here's the guy that invented PCR, which is a process that's used in the ion torrent. And it's polymerase chain reaction. It basically allows you to take a piece of DNA anywhere you like, as long as you have the sequence, and amplify it up. This guy here, Kary Mullis, was driving home one day in his car, and he came up with it. Just came up with it. It's eloquent. At best, it's just very smart. Guy got a Nobel Prize for it too, although unfortunately he has some interesting thoughts about other areas of science, which kind of put his favorability out of prospect. But anyway, yeah, well, there you go. But simply put, if you know the sequence, the DNA is denatured, just like in the genome, and then a primer is put down. It's called a primer. It's a short sequence of genomic DNA or DNA that you know, because you know the sequence, and then it just binds, and then it just amplifies up, and that's it. So you get a denaturing session at 94 degrees and annealing, because the primers have to lock. Then you mix it with an enzyme, DNA polymerase, which is exactly what happens in the genome, and then boom, it elongates. It collects the nucleotides and just puts it on. It just reads it and puts it on. Thousands of bases per minute. And basically, and I put some links down here that if you type in, you can get to see the videos that describe it going from just the single template to basically 2 times 10 to the 35, which is 68 billion copies in a simple 35 PCR reactions. For quantitative PCR, it's got a 40. I think some of this stuff does very large volumes of PCR. Oh no, it doesn't do that many. It does a small number. Has to, or else it gets bias. But anyway, that's really it. So it's very simply a case of copy, 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 and so forth. And this is what they used to do in Russia. In Russia, whoever's been to Russia. I was postdocing with a Bulgarian guy who used to do this by hand. So I drew it. I thought I got to draw this because that's what he did. He'd sit there with his tube, and he'd sit with a hot water bath, and he'd go from 55 to 72, and then put some more enzyme in, repeat it again, because the enzymes weren't necessarily very heat labile, until somebody found Taq polymerase, which is Thermophilus aquaticus, and that sits in the volcanic vents, and that enzyme therefore is what's used in here and has a longer lifespan, and is used now in everything that is PCR. All of these instruments are fancy. I've worked on all of these things. Crazy. But anyway, so you've gone from hand to instrument. This one here was a great one that came out because it was 4 independent heads, 96 well plates, and you could program it. This was big. This thing sold like hotcakes. It was amazing. Nobody could keep up with it. Everybody had those things. Okay. Oh, and this one here, sorry. This one here works by air. So this is just a block, a heat block. It's called the Peltier effect, or Peltier blocks, sorry. And you're able to cool it and heat it pretty rapidly. This one here uses air, the Kyogen. And it sits in a circle and the air just rotates around. And it cools it and heats it. Get the same results. Very clever. Primer design, pretty simple. It's remember I was telling you that the AT is a weaker bond than the GC, so therefore the GC is worth 4 degrees in this general rule, 2 degrees, and there's the equation that you can use to give you a 20mer. And then I just took, this is an EGFR, one of your genes. This is EGFR, one of the exons, and I designed some primers myself. I put 2 of them in, made 2 going this way. Everything goes 5' to 3', and then this is the exon that I wanted to be sequenced, or amplified, sorry. So I made 2, just in case, and I calculated that I made with 8 GCs and 12 ATs, and I have an annealing temperature of 56. So in the program, in the machine, I would put 56 degrees in for that temperature. Otherwise, everything else, 95 and 72 for denaturing and elongation would be the same. And so that's it. It just amplifies up. Now, there's a lot of software out there that does it for you. I did this by eye, and I know they're actually good. They'd actually work very well. But there's a lot of software out there, and in fact, there's a company called DNA Software if you're ever interested, and I did give you guys a paper around that. Making primers is it's more than just doing this equation. There's a heck of a lot more to it, and it's really fun. It's really a lot of mathematics, but it's all about thermodynamics, delta G, and how they all interact. And it's not, that's very basic, but if you understand that, then you get the kind of gist as to what's going on in the ion torrent, because you've got a whole load of primers that are going in to a reaction, that are looking at the panels, that are all amplifying up. So this is an example of just one gene, one mutation, sorry, one exon, which could potentially have a mutation in it, and that when amplified and sequenced, you'd see it. So detection of PCR fragments has really moved on from the good old-fashioned days of looking at it, putting on a gel, where you'd see either 2 bands or 3 bands if there was a mutation. It really depended on what you were looking at, and the percentage as well of wild type versus mutant. And you can see as you move down from wild type, from 0 to 100%, this band disappears. Quantitative PCR is a real nice way of measuring it through fluorescence, the presence of your product, and in particular for infectious diseases, knowing that you've got the presence of a pathogen inside you, your primers will detect that, and you'll see a nice one of these bands come up. And then this one here is where we're at now. Digital PCR is to take your tumor and ask very simply, hey, listen, is it heterozygous? Has it got heterogeneity in there? And so you can look at different mutations in the cells, and you can start to detect through this diagram here how many cells in your sample actually have the mutation. So you actually can quantitate it. It's called digital PCR. It's really super nifty stuff. Rain Dance Technologies has really made a big headway in there. And you can isolate one single cell, and it's put into an emulsion, very much like what goes on with the ion torrent technology. The DNA is put into an emulsion, oil emulsion. The solutions are mixed in with it, and the PCR occurs. And because you keep dividing it up over and over and over again to millions of PCRs all occurring at once, that then pass through, and because it's fluorescence, and then pass through a monitor that can measure the fluorescence, you actually get a number. You can actually determine whether you have in your tumor 20 cells that have one mutation, 1 cell that has one mutation of those mutations, or whether you've got 100 cells. It's really nice. It quantitates it beautifully. So expression profiling, you're going to hear this from some of your customers. This is really a little bit of what I talked about, sequencing the cDNA libraries. That's one way of doing it. As I said, you had 25,000 genes, 1 million proteins, and these alternative splice variants, these provide different expression profiles, different cell types. A gene that expresses a protein in one cell type will be expressing it at a different level in another cell type, because it has a value in that cell. It knows it, it's programmed to understand it and its value. When that modulation changes, that's when you get that sort of issues occurring. Why is it I can't walk out in the spring and not get affected by pollen, whereas somebody else can? Well, actually it's the other way around. I'm okay. I'm quite lucky I get away with it. But you get that variation, and that's because of all this expression profiles that you can pick up in each cell from each tissue. And the data that's out there in the public domain is huge and voluminous. It's amazing. You can look at RNA, through microarrays, or you can look at proteins through what's called ELISAs, which are enzyme-linked immunosorbent assays. And mass spectrometry is really starting to be another technology that could become a cool diagnostic tool. I'm not going to talk about that. Microarrays, it's closer to the DNA. Well, it is the DNA, so these drawings depict here. I was at a meeting. Ed Southern is an English guy who came up with the Southern blot. And very simply, that gel that I showed you with the bands on it, let me go back. You could take this and blot it onto a nylon membrane, and then you would hybridize another probe on it, and it'd be radioactive, and you'd be able to detect the presence or absence. And so this is called Southern blot, named after Ed Southern. Now, Ed Southern was at this meeting I was at one day, and he came up and he said, listen, I've got an idea. I want to tell you about it. Here's a here's a slide. It's a standard slide that you use on microscopes. And I've attached all these oligos on here. And you can actually take RNA that you've isolated, made it into cDNA, or just isolate the RNA, whatever, and then just hybridize it, and it will attach. And if you have some kind of color, you can actually detect the amount of that type of RNA that's there. So, wow, I'm sitting there going, I don't get this. But anyway, a few years later, everything's automated. It's huge. There's microarray slides. There's all this data. And everybody's into trying to understand how cells expressing genes at various levels during various times, during an animal's lifestyle, whether they were treated with something or without something. And very simply, if you took tumor cells and compared it with control cells, that's all you're doing. You isolate the RNA, you make it into a cDNA, as I said to you, which is basically back to, it's called C, stands for complementary. You convert it back to DNA because it's more stable. RNA is not very stable, and hence you'll hear about the formalin blocks. The RNA is not very stable in there. That's why doing it with the DNA is good. Now, some people are asking, ooh, can we try and see if we can get some of that RNA? But it's tough. So convert it quickly to DNA, complementary DNA, and then you label one all red, you label one with green, and you literally look to see the differences between the control cells versus the tumor cells. That's how the expression profile is understood between cells from different parts of the body. You could do it in a dish, you can grow cells up, treat them, do all sorts of things. And then there's all this computational biology that comes into it. Really exciting stuff, great publications, and like I said, you can go online and look at volumes of data that came out. Biopsies, pretty obvious. This one here, so if they're talking about biopsies, it's what the pathologist examines, and then of course he does it under the microscope, so he can get a good sense of that, what's there. Sorry about this gruesome picture, it's a lung tumor. So different types of biopsies. One's called an incisional, where only the sample of tissue is removed. That can be painful depending where they're taking it from. Excisional, where an entire lump or suspicious area is moved. Needle biopsy can be very painful again. It can be a wide bore, or very fine to get an aspirate, and then of course that aspirate's laid out on the slide, and the pathologist is able to look at it. But you can take that material and sequence that too. So it doesn't matter whether it's solid or liquid, it can all be sequenced if there's DNA in there. And then when you get to cytology, a little bit of histology then is really taking those formalin blocks, they're sliced and then they're stained with hematoxylin and eosin. Hematoxylin basically lights up the, the nuclei, and then eosin picks up everything else in different shades of red to purple. So you get a dark color, that's a nucleus, and then all these other stuff. And then you can look for differences between normal and tumor cells. And when looking at these things, this is endometrial cancer here, pathology says, oh, look at that, I can tell that's a tumor cell. So, and then recognizes it in relationship to the structure of the block, of the tissue that's been taken. Fluorescence in situ hybridization, FISH, if you ever hear of the word FISH, be aware what FISH is. It's basically karyotyping, which is basically taking your chromosomes here and then just coloring them actually. Again, same thing that's going on here. you've taken a piece of DNA, you've labeled it with a dye, and then the label is able to bind to the chromosome to give you different colors. And it's pretty, they call it painting by numbers in the early days, and you can look, I'm not going to go through this, but you can look at it and you can tell what pieces are missing, what's been inverted, what's a derivative, and so forth. So derivative, DER, del is deletion, and it's added to 9, and it's Q3. So really cool stuff. So remember fish, fluorescence in situ hybridization. And then this is an example of a test that uses fish, which is the, I mentioned earlier to you, which is the EML4 ALK gene, which run on different parts of the chromosome, or the same part of the chromosome, but they're on different strands. And so it had a flip, and now you've got a hybrid that's occurred, a fusion protein, and then this one here is a companion diagnostic that's come out, and so if you have, it's called pull apart or break apart, and so if you see that and that, then clearly you have the opportunity to go on the right drug. We talked a little bit about genomics and genetics. Just to reiterate then, when it comes to the testing component, genetic testing is when you're looking at variants within a gene, monogenic diseases, complex diseases, when you start looking at and measuring the interaction of all those genes with each other. The expression profiling is a classic example of genomics. There's just a ton of data and you're looking at all of them and how these genes are interacting with each other. So genetic tests then are diagnostic. You've got a diagnostic, so you've got a phenotype, and then you have a genotype. The genotype in this case would be, for example, a Down syndrome. A predictive genetic test would be BRCA1 or 2. Something that's pre-symptomatic that hasn't come down yet is DMD, Duchenne's. A lot of people are starting to look at, hey, listen, who's my partner here? Do they have something that I should know about? And I'm sure the partner's saying the other thing too. So having a what's called carrier detection, especially for cystic fibrosis. Of course, prenatal is big, big, big. And so trisomies 21, 18, and 13. And then the last one, the pharmacogenetic, really is just looking at the profile of your tumor, or even your, even as you stand today, and saying, well, you can take this drug. And some drugs like warfarin is a classic example. It's turned out that people have different responses to Tylenol and Advil based on their CYP genes, so CYP genes. So it's that pharmacogenetic, connecting your profile to drugs. All right, this is what you came for. It's the iron torrent. I don't know what you have in what goes on at HQ, but I'm assuming you got the full workup, every piece of toy that Life Technologies has to offer. Now, when I was at Life Technologies, they bought Iron Torrent for $350 million, and there was another $350 million on the table for milestones that were met. So that was a huge $700 million undertaking, and I can tell you the aggressiveness with which this company has worked at keeping Iron Torrent separate from the main company, allowing them to do what they had to do. I believe they've met some of that $350 milestone. So some people are very rich right now. From a simplistic process, it's very much you get your DNA, you put it into the one touch, it does library prep and target enrichment into the sequencing, and then this simple computer which has software in it basically will tell you what the readout is, and you get the report of the patient's results. And I'm going to take you through each one of these steps a little bit to give you a sense what goes into the chip. When you look at the first chip that came out, the PGM 314. The 314, it has 1.2 million sites, and I'll show you how those 1.2 million sites come together, with an output of 40 million bases of data. That's a volume. You know, I just showed you the Sanger sequencing. You'd be lucky if you got 200-300 base pairs. This is like 40 million, so it's huge. Runtime is pretty quick to get 40 million. There's no way you get 40 million doing the Sanger sequencing the old way. Good output per day. Now the average read is definitely, this is out of date because these guys just constantly keep updating it. There are 400 bases, and I think they're talking about getting up to 1,000. So you get the number of reads up to 0.6 of a megabase. And then the chip prices are down here with reagents and instrument price. This, by the way, is really deceptive because everybody goes, oh, $50,000 for an instrument, I'm going to buy one. Except you realize that you're going to need this toy and this toy. And that gets you to about 100 something, or maybe 95. So here's what the ion torrent can do. I've circled basically what it can do. You can look at these, the chips and what they can and can't do. The ion torrent is limited in its applicability, and its applicability is targeted, which is exactly what your panels are, targeted genes. So therefore, it's not like it can't do that. It can do it very well, and it can do it for all three chips. So here's the chip breakdown. So the number of base reads as you go out, and the output, the 314 that you have in your hands. So you can see that you've got 200 base reads in 2.3 hours, 400 base reads, 3.7 hours. If you want to push it out that far, it takes that long to get that type of...
Who would this information be relevant to? Who'd want to hear about this? Nobody. Okay.
This is for you to know, just in case somebody says, tell me about that chip.
Okay.
I'm sure somebody will, because they'll say, tell me about that chip, I'm confused. But this is stuff that's online, and by the way, this stuff gets out of date so quickly because this company just keeps updating really rapidly, every 6 months. Yes?
Well, I was going to ask you, can you use an updated chip in that same machine, or is it...
Yeah, this machine takes these 3 different chips. So this was the first chip, this was the second chip, this is the 3rd generation chip, but it's more designed to take more and more and more samples. And you can put different patient samples on. I'll tell you how you can do that. It's tagging them so that you have a barcode on the DNA. Again, this is really just for you to understand about the read qualities. Q numbers, it's a Q20 to Q30 represents the quality of the base that's called. Remember that because it's important. We talked about, Pablo and I were talking about some sequence that comes out and it's not sure what it is. So it's all about quality. So over a stretch of 100 base pairs, 200 base pairs, the aim is to keep this quality of a base call, which is that nice peak that I showed you in some of the other ones, the chromatograms. Between Q20 and Q30, this is a number that's given to the quality of the base, that it falls between 99 and 100% as you go out. And you can see as you go out, and this is what they're getting better at, is keeping this up and up and up and up and up and up as they go to 400 and 800 base pairs. Now, one of the difficulties with this type of sequence is what's called homopolymer stretches. These are the same base, A, going out. It gets harder and harder for this type of technology to manage, and typically they have found that 5 is really of relevance towards disease versus relevance in the human genome if you're sequencing the whole genome. So it does a good job going out to relevancy to 5 for disease, but I would suspect that it has, still has problems when you get large runs within the human genome, which resides within introns for sure, that it cannot get accurate. And that, by the way, plummets quite dramatically. So from an all-round accuracy, it does a good job. The key to the accuracy is the way the software interprets that data. Before we get there, let's look at the workflow and how a sample comes into the lab and gets processed. So there's sample preparation starts off, you go to library preparation, then you go to the sequence, and then the data analysis. So the DNA comes from a formalin fixed, paraffin embedded slice. You may get the whole block maybe, but it's usually a thin slice. And then there are kits that will allow you to isolate the DNA from that formalin fixed paraffin embedded block, because it's not, it's a preservative. It's all been preserved to last forever. And so the kits that I believe Life Technologies recommends is like PureLink Genomic DNA Mini Kit. And once the DNA is isolated, then it goes into the library prep. You get PCR amplification of the DNA with the target specific primers. So for every panel, there is a set of primers that will be put into that DNA that has been requested by the doctor to have tested. So the doctor says, I want a colorectal panel. You take all the primers for all those genes, you throw it in with the genomic DNA, amplify it up as I showed you through PCR, and then you basically go into the sequencing. when that's done, and you ligate sequencing adapters to that. And I'll show you some pictures, but basically you've got these beautiful pieces of amplified DNA, and you're just going to glue on another set of primers that will allow you now to sequence, and you can just sequence straight through that. And I'll show you some diagrams that will really help register what that means. And then finally, the software basically takes all that sequence that comes through and then just lines it all up. And there's a lot of depth, and as it sequences across, based on the primer design for looking at certain parts of the genome or these genes. Maybe you're just after that example I gave you with EGFR1. One mutation within an exon, then in case it's going to be looking like this. If it's a big exon of about 1,000 base pairs, then you may have to do staggering across to get the full sequence. It really depends on how those are designed. That's your proprietary technology, I would suspect, and that will give you the results that you're seeking through this setup. And you can see the time. That's why when you turn around and say, we can get you results within 3 days, you can see that the time it takes is probably about a day. And Life Technologies does love to show off that compared to some other people. So theoretically, with little hands-on, so you're talking about a day, and you get the result, and then it gets obviously reviewed in-house, and then gets put together in a form, and then sent out. So 3 days is a really good turnaround time. And by the way, I'll show you tomorrow, there are very few, if any, companies that I could find that could beat 3 days. So you guys are good shape there. That's really good. And by the way, I think 3 days is really good. I think it's a tough one when you have to wait. And some of these companies were like 16 weeks. Oh, you got cancer here, right? 16 weeks. So 3 days is great. Okay, so I know it's a little small, but here's how you make that template to go into the sequencing. So there's a little bit of technology in here, so work with me as I take you through it, but stop me if you get a little if I lose you. So simply put, you get your DNA up here, and then it gets broken up, and then there's in this case, the piece of DNA that you're interested in is isolated away from all the genome. So then stuff you're not interested over here, all the stuff you're interested in here. So now you get a PCR reaction that's clean. No false amplifications coming in, and any other types of reactions that could possibly occur. And then there's a bead capture, so after the cleanup, and then you do your amplification, as I said, and then you go into sequencing. On this kit, you have it slightly different. You actually PCR everything up, and then you go in with a bead or some kind of capturing agent to take out all that PCR amplified product. So it's another way of doing it. So even if you get mispriming, mis-PCR, this primer will only bind to the product that's amplified, clean it up, take it away from everything else, and then you go in. It's very much a simple case of how do you enrich? Do you enrich before PCR, or do you do it after the PCR? That's really it. And then this is this company, Raindance Technologies, where the primers, where it all comes together. You see here, you've got the primer library, so that's a colorectal panel with all the primers in to PCR amplify all the regions in there, and your genomic DNA. And then it's really clever the way they put this together. You get this kind of oil emulsion technology, and it's passed through together, and the liquid for the primers gets pushed into the these oil emulsion drops, and it goes down, and then it comes out ready to get sequenced. And you can have in a tube, I'm not joking, you have a tube like millions of these little drops in, you just put the one tube into the PCR machine, and that's it. It just all does it in there. And then once it's done, you take it back out, you put it back into the machine, and as I mentioned to you, it then reads which ones have been PCR'd. But in this case, you're going to take those droplets, and you're going to sequence each one of those on that chip. Because you've got 1.2 million wells, you've got 1.1 million sort of these droplets. Each one of those droplets would file in there, and you'll be able to sequence each one of those pieces of DNA. So it's really about enrichment and how it gets there, and I've got some more figures. And then barcoding. Right here is the key, it's a take-home message. That barcode is unique. That tells you that the patient who's come in, if you haven't already tagged it with a barcode, then you've got another layer of barcoding at the DNA level, so that when it's lined up, then the sequence comes through, you know that that sequence that you already attached onto the patient's genomic DNA is unique, and that barcode is one way of keeping the indexing tight. So you've got sample comes in, you barcode on the tube, but you can also barcode in the DNA if you need to go that far. Again, I don't know exactly the processes that are being used, but I did want to just show that there's a couple of ways of really tracking the patient sample through the system.
Say a patient gets diagnosed with colon cancer and then they find out it's spread or something else. Do they need another sample, or can it be reprimed?
Depends on how much of the genomic DNA is left. You might want to go back to the FFPE block, the formalin fixed. The doctor may say, look, I've got a patient who's got metastatic cancer, looks like it's in the lung and in the colorectal, in the colon. So at that same time, you can ask with that DNA. So you only need 10 nanograms of DNA. It's a really small amount to get all this information out. So again, I think it's really how the process is put together in the lab that question's best answered.
I would say that 99% of the times we're going to have to keep the DNA that we receive, the tissue that we receive, and with that we need to preserve it for X amount of years.
Right.
So if for any reason whatever we tested somehow derives in something else, we could retest it without getting another tissue.
Without getting another tissue.
Most probably, I would say pretty much all the cases.
Okay, so as I mentioned to you, the bead is the one that captures all the DNA, and the enrichment's on the bead. This bead drops into the wells, one of those 1.2 million wells that are in your chip, and then off this comes really that Sanger sequencing that I talked to you about. Except one important thing, it doesn't measure what nucleotide is put on through some kind of color or radioactivity. There is, as part of the reaction, a release of a hydrogen ion, hence the ion tolerance. It's a hydrogen ion, and it's all about measuring this H+. And you can see here quite nicely that once when the base is put on, a hydrogen ion is released. So in each well then, there will be flooded into it one base. So if it turns out that it's a G and you flood in the C, then the polymerase can take that C and attach it. And in doing so, an H+ ion is released and it's measured down here. If there is no, if it wasn't a G, say, well, still a G, but say you put an A in, then it can't move it forward. Therefore, no ion is released, and you will see it not come up on here as a signal. It's as simple as that, and that chip that you have has got multiple layers within it. It's got not only a microfluidic chambers in there where liquids are passed in and out all the time through the machine, but also the sensor that detects the hydrogen ion is so sensitive that it's able to understand which, in this particular well, which hydrogen ion has been released, or for a base, and so forth. So it's a very complex machine. There's more layers to it than meets the eye, but from a very simplistic standpoint, it's all about measuring the hydrogen ion. And if you can get that bit, and if you are able to, if you can digest that one component, which is when a nucleotide is added on, a hydrogen ion is released and it's scored, that's how it's basically done. It's just done, and there's like 1.2 million of these reactions going on at once. So data analysis is, again, you can create any process you want so long as it's validated and it's appropriate for bringing the result to the table that goes into the document. But simply put, the data that pours off is then imported into the machine, it's analyzed, and you look for the variance, and it stacks it to look for the there's a base, there's a software that calls the base against a reference. So there's always a reference in there. That reference comes from the sequencing of the human genome, and then your patient sample is compared against that. And if there's a change, a difference, a variant, whether a different mutation, as in a different base, an insertion, deletion, against that the reference against the reference, sorry, against the reference DNA, then it will call it, and then you'll be able to take that result and put it into your report. And then tomorrow, I believe, Dr. Cohen is going to talk about the report, so I'm not going to address that today, but just know that this is what occurs in that big magic box. And then, of course, all this data is archived. Again, I mean, Pablo, you might want to address how the data is stored and kept, because that's going to be a question that your customers will ask. What happens to this data when the results come out?
So basically what we do with the lab, we take all the lab process, and we got all the information. That information goes through the web to the mainframe of Life Technologies. They process that information. I return that comparison that Richard was mentioning. From that, we take all the information and this is a very, very tedious and difficult process, because it's not just a button that you push and it says mutation, deletion, or whatever. It gives you, in order to make it simple, it gives you like a geographic position or whatever you need to determine, and you could take that information from another database we call Cosmic, and from that Cosmic database it's going to tell you exactly the mutation and the location and what are and then from there, from knowledge and experience from database again, you could I don't want to say propose, but just refer or bring out information about possible outcomes of that mutation. That is not just saved in our lab, it's also saved in Life Technologies, and there's many backups that you do for that, not just physical, like paper-wise, also file, and I would say that's conclude the part of the archives, but and then the report, of course, is delivered to the patient. We're going to talk about the report in length tomorrow, but that's pretty much that area.
Great, that's perfect. I didn't know that system, so that's everybody, every lab, I'm sure, has its own way of dealing with that. But that's kind of important to know, because that's always going to be something that what happens to this data. So make sure that you understand that well. Yeah.
When you have the sequencing and you get the results back and it says the chance for the gene of expressing itself is 34% or whatever the variable is, how is that actually coming up with that number? Is that the actual sequencing, how many it takes to find that gene, the percentage?
I don't think that Pablo and the team are using that. It's simply the reference sequence, and do you have that mutation or not? Simple as that.
Exactly. The information, that's why I was very, very...
because I've seen some of the
I like
PCR
to
tests that do
reiterate,
that, and I just didn't know how they do that.
I understand where you're coming from.
Right.
We discussed this in the previous meeting, and we were talking about the chances of this and the
Right,
chances of...
percentage, right.
We don't deal with chances.
Right.
We deal with black and white.
Right.
It's either there or it's not there.
Right.
And then whatever it's going to happen after that, it's not us.
Right.
Just the clinician is going to take everything in consideration, and he will assess what are the things that could happen or not. We don't have any chances, no percentages, no remission, nothing. We just say there is a mutation. This is the location of within the DNA.
Right.
This is the gene and the mutation is this, that, and that. And because of that, you do whatever you need to do.
Mm-hmm.
No percentages, no...
And go back to the earlier slides, homozygous, heterozygous. Are you homozygous wild type? Are you homozygous carrier or with the mutation? Or even heterozygous, where you have the wild type and also the mutant? So that's it. And that's a clean, that makes your job easier, because you don't have to deal with any of that stuff.
I'm just thinking for comparison for some other tests, because there are the PCR tests that do that. So then when you say, well, you have the gene versus a company that then is going to say, well, yeah, you have the gene, but it's only 34%. That's what I'm trying to say. Like, how do you, if you're testing similar panels, how is it going to be just saying that you have it versus maybe the percentage? And then how do they even get to that?
So the only way I'll address that is two things. One, I think for cancer panels that other companies use, they're making the same calls as Pablo and the team are, which is very simply, you have that mutation, where you were